Hi everyone, my name is Cruz and I'm an undergraduate student at the University of Massachusetts Amherst. I'm currently performing an honors thesis with Dr. Alex Gerson and Corey Yalow in the Department of Biology. My thesis is titled Digestive Assimilation is Body Condition Dependent Following Long Duration Flight in a Migratory Bird. So, as you may know, each year many birds undergo seasonal migrations as they travel from breeding grounds to wintering grounds in order to take advantage of changing resources throughout the year. And these long-distance flights are really energy-intensive. They require birds to operate at a really high metabolic rate, and they can also last for hours. So in birds such as songbirds, which are the birds used in this study, they will fly all night and only stop during the day to rest, whereas some shorebirds can actually fly for days on end. So you can imagine that it takes a lot of fuel to power these flights. And the primary fuel that birds use during flight is fat. Before migration, birds become hyperphagic, so that they increase their intake, and they do this so that they can build up large fat stores which can be used as fuel. But in addition to fat, birds also burn protein, and this protein catabolism contributes 5 to 15% of the total energy demand during flight. There are several reasons why birds burn protein, but one reason is for metabolic water. When protein is broken down, it yields more water than when fat or carbohydrates are broken down. So, for this reason, it's been found that under dry conditions, birds will increase their rates of protein catabolism, and this allows them to offset water loss during flight. So during flight, birds lose both a lot of fat and lean mass. And you can see fat mass loss in these images. So on the right, you can see a bird before flight. The feathers have just been parted, so you can see fat deposits under the skin. It's kind of like a yellowish color. And then on the left, this is after flight, and all of the fat deposits have been lost, and it's really just muscle and bone. So due to the large protein catabolism during flight, this leads to a reduced mass of many internal organs. And this disproportionately affects the digestive system. So organs such as the intestines and liver can actually decrease in mass by 50%, which is a lot. And you'd expect that this would affect digestive function. So one thing that has been seen is that when birds stop at stopover sites to refuel and rest, they have a lag in mass gain. And one reason that has been hypothesized for why this happens is that if birds have catabolized a lot of protein and their organs, such as their intestines, are physically smaller, they might just physically not be able to intake enough food to gain mass at the rate we would expect them to. So you can also imagine that birds that are breaking down more protein during flight will have an even greater reduction in digestive organ sizes, and this will affect digestive efficiency. However, studies have also found that birds can compensate by increasing the retention time of food in their gut, and this allows them to maintain digestive efficiency. So retention time is the amount of time that the food is held within the digestive system. So if the food is spending more time within the intestines, for example, they're going to be able to absorb more energy from that food. This is important because the events that occur during stopover can affect the success of migration as a whole. If birds lose more lean mass, rebuilding this lean mass is going to take a lot of time, and this may increase the length of their stopover. And if stopover is longer, migration will be longer, and this means that they will arrive at their destination later. And because of this, they might miss out on optimal breeding resources such as territories, and this will decrease their overall fitness. So in this study, we used yellow-rumped warblers, which are a migratory warbler, and they were flown or rested in a climatic wind tunnel under different humidity conditions. So these are high evaporative water loss, which is a drier condition, or low evaporative water loss, which is a more humid condition. And so there's a video showing a bird flying through a wind tunnel, and this is really voluntary. The birds can stop if they want to, and this would end their flight, but a lot of birds choose to fly for up to 6 hours. And then before and after flight, quantitative magnetic resonance body composition analysis is performed. This is abbreviated QMR, and this measured changes in fat and lean mass during flight. So when this is performed before and after flight, we can see changes in body composition that happen during flight. And then for 3 days pre-flight and 3 days post-flight, data was also collected on food intake and excreta production. So the amount of food that the birds ate and the amount of excreta that they produced was monitored, and then samples of food and excreta were collected and frozen. And then back in the lab, these frozen samples were put into an oven and then dried out until only dry mass remained. And then the gross energy content of the food and excreta samples were determined using a bomb calorimeter, which is the machine pictured at the bottom of the screen. And this machine basically determined the caloric value of these samples. And using this value, we could calculate the metabolizable energy coefficient, or MEC. So this equation subtracts the amount of energy in excreta from the amount of energy that the birds ate, and it determines what proportion of the dietary energy that the birds utilized. So using these values, the digestive efficiency was evaluated pre- and post-flight. So getting into results, there were a total of 27 birds in this study, 13 flight birds and 14 rest birds. And as we expected, the birds that flew lost more mass than birds that were rested. So in this figure, you can see flight duration plotted against mass loss between the flight and rest birds, and the birds that flew lost more mass at a higher rate than birds that were rested. And additionally, fat mass was also lost during flight, and it followed a similar pattern as mass loss. So it was higher in flight birds and increased with flight duration. And in addition to fat, birds also lost fat-free mass. So you can see in this figure, the drier condition is in red and the more humid condition is in blue. And what you can see is that birds that flew under drier conditions are losing more fat-free mass than birds that flew under more humid conditions. But we found that humidity treatment did not affect fat loss, only fat-free loss. So once we examined the changes in body composition, we could look at how these affected digestive assimilation. And we wanted to do this by comparing values pre- and post-flight. So what we did was set the day before flight as like a baseline value, and then we calculated the change in these values post-flight. So that is what a lot of these graphs will represent. So if you look at the bottom left graph, this is showing fat loss versus food consumption. What you can see is that as fat loss increases, food consumption also increases. But if you look at the graph on the right, this is showing fat loss versus the change in excreta energy content in calories per grams. So these birds were eating in a diet with a stable caloric content, which means that if the energy density of their excreta is changing, they are absorbing either more or less food from the excreta, more or less energy from the food that they are taking in. So when you look at this graph, although there is a very slight downward trend, the energy density of excreta does not significantly change with fat loss. But when we look at fat-free loss, it is a different story. So the graph on the bottom left is showing fat-free loss versus the change in excreta energy content once again. What you can see is that energy density of excreta is decreasing as fat-free mass loss increases. This means that these birds are extracting more energy from their food. But if you look at the graph on the right, this is plotting fat-free mass loss against the change in food consumption, and this graph is pretty flat. There's no change in food consumption when fat-free loss increases. So in addition to these changes between different body compositions, we also saw changes in humidity treatments. So if you look at the figures at the bottom of the screen, these are plotting 3 different values pre- and post-flight between humidity treatments. And the x-axis is showing day number. So day -1 is the day before flight, and day 2 is 2 days after flight. So if you look at the graph on the left, this is showing the change in food intake. And what you can see is that both humidity treatments increase their food intake after flight, but the birds that flew under drier conditions increased their food intake at a greater amount than birds that flew under more humid conditions. And then if you look at the next graph in the middle, this is plotting the change in excreta in calories per grams. And what you can see from this graph is that birds that flew under more humid conditions in the blue are increasing the energy density of their excreta, whereas the birds that flew under drier conditions, which is the red, are decreasing the energy density. So that means that these birds are absorbing more energy from their food. And then the graph on the right is showing the change in MEC. And MEC, just a reminder, is the proportion of dietary energy intake that birds are absorbing. So it basically represents digestive efficiency. And what you can see is that birds that are flying under drier conditions are increasing their digestive efficiency, whereas birds that flew under more humid conditions are decreasing digestive efficiency. So these changes might be due to differences in body composition between the 2 humidity treatments. As you may recall, the birds that flew under drier conditions lost more fat-free mass than birds that flew under more humid conditions, so this may have caused the changes in digestive efficiency and energy absorption. So to summarize everything that I've gone over, post-flight refueling is different depending on body condition changes. So birds that lost a higher amount of fat are increasing their food intake, whereas birds that lost a higher amount of fat-free mass are absorbing more energy from the food, leading to a lower energy density of excreta. Additionally, environmental condition is also affecting refueling strategy. So birds that flew under drier condition, which lost more fat-free mass, are becoming more are increasing their digestive efficiency after flight and absorbing more energy from their food, whereas birds that flew under more humid conditions are decreasing digestive efficiency and absorbing less food from their excreta. And lastly, birds don't reach their physiological limit in this study. So even though they flew for up to 6 hours and lost a lot of fat mass and fat-free mass, these birds don't limit their intake after flight, which means that they don't reach their physiological limit in this study, even if they lost a lot of fat-free mass. So these are my references, and I just wanted to give a special thanks to former and current members of the Gerson Lab, including Dr. Alex Gerson, Corey Elo, and Derek Groom for all of their help and support throughout this project. Thank you so much.